through mitosis or through interphase. When we're talking about meiosis, there are two cell divisions to meiosis, right? There's meiosis 1 and meiosis 2. What are the stages of those cell divisions?
Prophase, metaphase.
Try again. Prophase 1, metaphase 1, anaphase 1, telophase 1. And then there's prophase 2, metaphase 2, anaphase 2, telophase 2. Where's the prometaphase? There is none. Okay, that's because prophase 1 is actually divided into 5 subcategories, 5 substeps. So,
Would you
anyway,
know prometaphase for your test?
no. Actually, I think I do have it in there in one question, like, because it's in your book. Anyway, okay. So we've got our nucleus here, and our chromosomes. We've got a cell with 6 chromosomes, right? All right. And we go through G1, right? S. Our chromosomes are going to replicate. So we make identical copies. How many chromosomes do I have now?
6.
I still have 6. Absolutely. How many chromatids? 12. Okay. So now I go through my G2, checkpoint, check. Yes. Okay, so we're good to go. We enter into meiosis, prophase 1. There are 5 key things that happen during prophase 1. What are they? Let's set up our centrioles. We replicate these guys. Nope, that happens during D2. I forgot about that. So what happens during prophase 1? Centrioles move to the opposite poles, right? Move to the opposite poles. And they put out spindle fibers. What else happens during prophase 1? The nuclear envelope will disappear. What else happens during prophase 1? What happens to my chromosomes? They condense down. They coil up so you can actually see them, right? So those are the exact same things that happen during prophase, happen during prophase 1. Centrioles move to opposite poles, put out their spindle fibers, nuclear envelope disintegrates, chromosomes condense. The 2 new things that happen during prophase 1 are unique to prophase 1 and only happen during prophase 1. What are those 2 things? What happens with these chromosomes that only happens during prophase 1?
Crossing over.
What has to happen first? Synapsis. The chromosomes synapse. We have 2 chromosomes, one from mom, one from dad. They are identical chromosomes other than one came from mom, one came from dad, and, you know, blue eyes versus brown eyes or whatever, okay? I can roll my tongue. I can't roll my tongue. Whatever. I've got 2 chromosomes. These are called homologous chromosomes, right? My homologous chromosomes will pair up or go through synapsis, okay? They will pair up. Now, these 2 are not homologous, are they? They don't pair. These 2 are not homologous, are they? They don't pair. These 2 are synonymous, or homologous. Look how easy that went together. It's like they knew, right? These 2 are homologous, and they'll go together. So, homologous chromosomes pair up during prophase 1, and only during prophase 1. Once they pair up, now what can happen? Crossing over can occur. Which crossing over is an exchange of genetic information between non-sister chromatids. These 2 crossed over, it doesn't make a difference, does it? But I can exchange information between the mom and dad chromosome and get something completely different, okay? So, crossing over, I'm going to do this. Crossing over can occur like this, right? Where that exchange genetic information. But guess what? If it's long enough, it can happen again, all right? And so crossing over can happen back. So what was once on dad became on mom, and now it's back on dad again, okay? On that chromosome there. And the further apart the genes are, the more likely crossing over is going to occur. That's another thing. The closer together the genes are, the less likely crossing over will occur. All right, so we get this non-sister chromatids crossing over. Right? Same thing here. Non-sister chromatids crossing over. Exchange of genetic information. This only happens during prophase 1. It does not happen any other time in our cells at all. At all. It'll happen at least 80% of the time in all of our cells. That's not right. Oh, I hate these popbeads. They hurt my fingers. Okay, so prophase 1, chromosomes condense. Centrioles inside the centrosomes migrate to opposite poles. Nuclear envelope disintegrates. Synapse between homologous chromosome occurs. How many sister chromatids do I have here now?
3
No.
or 4?
In this right here. How many? Right here. How many sister chromatids are there? 4. This creates what's called a tetrad. Okay? This is a tetrad. The synapsis occurs, crossing over takes place. Only during prophase 1. No other time. Got it? What happens during metaphase 1?
They line up with the equator of the cell.
How do they line up? The homologous chromosomes line up completely random on the equator of the cell. Completely random. Now, this shows the yellow on top and the yellow on the bottom for these two, right? Completely random. Completely random. Now, we have 23 pairs of chromosomes. 23 pairs of chromosomes will randomly line up on the equator. 2 to the 23rd power is over 8 million possible ways that these chromosomes can line up. There's 3 types of chromosomes here. So this is 2 to the 3rd power. There's 8 possible ways they can line up here. They can line up like that, they can line up like this, they can line up like this, they can line up like this, they can line up like this. Completely random. This is what's called independent assortment. Well, it's not realized until anaphase 1, but this complete randomness of coming here on the equator is happening independently of anything else, okay? So it's just, that all these chromosomes are going to line up on the equator, completely random. Homologous pairs line up on the equator. That's what's different between mitosis and meiosis. Metaphase and mitosis, the chromosomes line up individually on the equator. In metaphase 1, homologous pairs line up. So I have 6 chromosomes in my cell right now, still, right? What happens during anaphase 1? Where do they pull apart?
Synapse.
The synapse during anaphase 1, the homologous pairs separate where they synapse together during anaphase 1. Very different than anaphase of mitosis. How many chromosomes do I have?
3.
I have, how many chromosomes here right now? 6. In mitosis I had 12 because the chromatids separated during anaphase. Here the homologous chromosomes separate, so I still have 3 chromosomes going north, 3 chromosomes going south, right? I reach the pole. Now, what kind of cell is this going to create at the end of telophase 1?
Haploid.
These are haploid cells. Why are these haploid cells?
So they all have pairs, sister chromatids.
Because they have sister chromatids, but they do not have a homologous pair to match up. They don't have that second chromosome that it was paired with before. So now I have half the number of chromosomes I started with. I started with a cell with 6 chromosomes, and now I'm going to end up with 2 daughter cells that have 3 chromosomes each. And each one of these daughter cells is different, isn't it? Because here I've got a long red with a little bit of yellow, medium red with a little bit of yellow, and a short red with short yellow with one little red. But just the opposite here, I've got this medium yellow with a little bit of red, long yellow with red, medium red with a little bit of yellow. Okay? They are completely, totally, and uniquely different. But I'm not done, am I? This is meiosis 1, where I take a diploid cell and go through cell division and create 2 haploid daughter cells. But I still have duplicated chromosomes. have to separate out these duplicated chromosomes. And in order to do that, I start all over again, right? We go into a time period, it's referred to as interkinesis. Don't really need to worry about that. But during interkinesis, one of the only things that really happens is our centrioles replicate again. So now I have two daughter cells that are not going to be big enough to do this in, but I'm replicating my centrioles here, like this, right? So I replicate those, and I have my chromosomes inside of the nucleus. I have 3 there, and I have 3 here, right? And so I go into meiosis 2, prophase 2. What happens? Everything happens again. Chromosomes condense, spindle fibers, centrioles move to opposite poles, spindle fibers are released, right? We kind of have all of this, you know, they're down here with their spindle fibers. Chromosomes condense, you know, they start to get pulled into a direction. Nuclear envelope disappears. Go into metaphase 2. Metaphase 2, they line up on the equator, right? Anaphase 2, they separate at the chromatids. Now, how many chromosomes do I have in this cell? 6. How many did I have to start with? 3. Okay. How many did I have to start at the beginning of meiosis? I had 6 at the beginning of meiosis. During anaphase 1, when my homologous pairs separated, I still had 6. Now during anaphase 2, my sister chromatids separate, I still have 6. Okay? When I get to telophase and cytokinesis and I split my cell off into 2 daughter cells, I now have 2 completely unique daughter cells here. The same thing here. These guys line up, they separate, I create 2 daughter cells. Where'd my chalk go? Right? 2 daughter cells. I now have 4 haploid daughter cells. 4 cells that have only 3 chromosomes. And each and every one of these 4 cells is genetically unique. Yellow with no crossing over. Yellow with a little crossing over. Red with a little crossing over. Red, no crossing over. Yellow, no crossing over. Yellow with a lot of crossing over, right? So, and then the same thing here. Red with some yellow, yellow. None of these chromosomes, not a single one of them, are identical in any of these cells, are they? So what was once all dad and all mom is now a mixture of your mom and dad that's going to show up in your own kids. Because this is now your egg or your sperm cell being produced that's going to be fertilized. Because you were a combination of your mom and dad. And now you take your mom and dad's DNA and you mix it all together. Ever wondered, well, you guys are all so young, but my sister's son is a spitting image of my father. Spitting. He's 7, 8 years old. And my dad, through the first 8 years of his life, you look at pictures and you look at my sister's son, they are identical. What do you think? She had probably a lot of dad's DNA and not so much of mom's DNA whenever that went into that son. And her husband, I don't think he even existed. And no, I mean, honestly, he is identical to my father. He's definitely not as ornery, but identical in appearance. Okay? So, these are 3 or 4 unique daughter cells that happen. Meiosis 1, prophase 1, chromosomes condense, spindle fibers form, centrioles migrate to opposite poles, synapsis occurs, and crossing over. Metaphase 1, homologs line up on the equator,